Welcome to it's Biochemistry, and this time we'll be discussing amino acid catabolism. Now, amino acids are involved in a number of chemical reactions, including peptide bond formation. But in this lecture, we'll be concentrating on the catabolism of amino acid, which lead to production of ammonia, and through ammonia, production of urea. So that is obviously urea cycle, and the urea cycle we'll be dealing in the next lecture. And compared to, let's say, carbohydrates or fatty acids, amino acids do not get a proper share of the discussion, maybe because amino acids give around 10 to 15% of the total energy need of the body after catabolism. This is unlike glucose or fatty acids, give the bulk of the energy need of the body. As also, the amino acids cannot be stored in our body, unlike glucose, which can be stored as glycogen, or fatty acids, which can be stored as triglycerides or other lipids. Amino acid catabolism actually involves here transamination and oxidative deamination, which lead to production of ammonia in liver. And as also, we consume actually lots of D amino acids, but through plants and plant products. And in our body, biomolecules dominate in L amino acids. So there is a mechanism that is D amino acid oxidase, the enzyme, which is involved in conversion of D amino acids to alpha ketoglutarate. And alpha ketoglutarate can be converted to L amino acids or can be channeled to TCA cycle for production of ATP. Now, let us start with the transamination and go to oxidative deamination. Now, transamination actually involves the transfer of an amino group from an amino acid. The actual definition goes like this. It's the transamination involves the transfer of an amino group from an alpha amino acid to an alpha keto acid. So this is the keto acid. You can see the C double bond keto group there. So an amino group is transferred to an alpha keto acid so as to form a new amino acid having the carbon skeletal structure of this particular alpha keto acid. And what happens to the original amino acid, the donor of amino group? It gets converted to a corresponding keto acid. So here again you can see the keto group, C double bond O, keto acid, and the same carbon skeleton is this one reaches here. Now this is catalyzed by transaminases, a group of enzymes which are also called aminotransferases, and aminotransferases is the more commonly used term nowadays, and it involves coenzyme also, pyridoxal phosphate, which is being used by all aminotransferases. And you can just go through the description, and these are two examples. You can see here one alanine and aspartate, two examples. Now, almost always the keto acid that accepts the transferring amino group is the alpha ketoglutarate acid here, and the glutamate formed, this amino acid formed can be used for the synthesis of non-essential amino acids. So this is actually a method for synthesis of non-essential amino acids also, apart from catabolism and the release of ammonia. But if non-essential amino acids are there, they are not needed afresh, this glutamic acid. Obviously there are two COH groups here, glutamate or glutamic acid, and the glutamate formed, we have a quick way to convert this glutamate into alpha ketoglutarate acid and release ammonia in liver. So that we will see in a moment. Now, here the enzyme involved is alanine transaminase. This is aspartate transaminase, and both of them uses pyridoxal phosphate as the coenzyme, which is derived actually from vitamin B6. And the pyridoxal phosphate initially acts with the donor amino group, amino acid, and accepts the amino group of that amino acid before it actually transfers it to keto acid carrier. So that is the mechanism, and these two examples, ALT and AST, they have got clinical significance also because liver cells have high activity of these enzymes, and any sort of cell death of liver, that is hepatic necrosis, due to various reasons, the level of these enzymes raises beyond the baseline level, because if there is an injury to hepatic cells. Now, in most of the cases, the new amino acid formed is glutamate, because we have got a mechanism to get to convert it into alpha ketoglutarate with release of ammonia in liver. Now, this process is called deamination, and of course there are different types of different mechanisms of deamination, all of which give rise to ammonia as the product. But here we will discuss oxidative deamination, which is the process which follows transamination so as to form or release ammonia. Now, oxidative deamination. Now, oxidative deamination actually takes place in two steps. First one here, and then hydrolysis here, and both these steps are actually catalyzed by glutamate dehydrogenase, an enzyme which is present in mitochondrial matrix. Now, the enzyme is unique in that here in the first step it can use both NAD plus and NADP plus. In fact, the forward reaction is favored. The forward reaction favors NAD plus as the coenzyme, whereas the backward reaction here, this one, this is a reversible reaction. So the reverse reaction actually favors NADP plus as the coenzyme. And what is formed is alpha aminoglutarate. You can see the amino group from here, it reaches here, and this is often termed as imino group. But this is an unstable compound here which gets instantly converted to alpha ketoglutarate in liver. And this is by a hydrolysis step with release of ammonium ion. And this alpha ketoglutarate, which gets released in liver, it can be channeled to TCA cycle, and it can be used for production of ATP energy. And after dehydrogenation, ammonia is formed, but this ammonia is removed very fast so as to drive the reaction forward. And as also high GTP or ATP acts as an allosteric inhibitor of glutamate dehydrogenase, whereas high GDP or ADP acts as an allosteric activator of the same enzyme. This is because glutamate will provide alpha ketoglutric acid, which will later provide energy. And obviously, if energy need is there, proteins can be broken down to give amino acids, and which can be later can be converted to or transformed to alpha ketoglutric acid and later energy. So that is a strategy followed by cells as far as protein breakdown and release of ATP is concerned. So this is the key molecule here, alpha ketoglutarate, which can be channeled to TCA cycle. Obviously, it forms from proteins originally, then amino acids, and which all of them gets converted to glutamate. So release of ammonia is there, and the prospects of forming energy also is there. Now, what we have discussed so far is what happens in liver cells. In other cells also, like peripheral tissues, including muscle cells, transamination and oxidative deamination take place with the release of alpha ketoglutric acid and ammonia. Now, obviously, alpha ketoglutric acid will be utilized by these cells for energy needs to channel into TCA cycle, but ammonia has to be taken up by liver cells for urea synthesis or urea cycle. Now, here, tissues like muscle cells leave the burden to liver here. What happens is that the ammonia, it's highly toxic. It cannot be released to the bloodstream, because if it reaches vital organs, including brain, which can give rise to cerebral edema and other issues, so it cannot be released as such. So what happens is that in peripheral tissues, including muscle cells, this ammonia combines with glutamate to give rise to glutamine with the expenditure of ATP and in the presence of the enzyme glutamine synthetase. Synthetase obviously utilizes ATP here. Now, glutamine is as you know, it's an amide amino acid having two nitrogen in it here, one amino group and another one here. So it's an amide amino acid, glutamine. Now, what happens to glutamine? Glutamine, it is harmless. It's an amino acid. It is harmless. It is not toxic. It can pass through the the bloodstream and reach liver. And in liver, what happens is that, so this is what happens in peripheral tissues here. Yeah. Peripheral tissues. Glutamate is converted to glutamine with the glutamine synthetase with expenditure of energy, and ammonia is taken up. And in liver, what happens is that the reverse of the process takes place with the help of another enzyme, namely glutaminase, which is present in liver. So glutamine gets converted back to glutamate with release of ammonia, inside liver mitochondrial matrix. And of course, this is important here. The ammonia is actually formed inside mitochondrial matrix. That also is a protective mechanism, so that ammonia can be processed there itself. Now, apart from this, muscle cells, for example, has got another strategy. This is called Glucosealanine cycle. Now, obviously, glucose can be converted to pyruvate through glycolysis. Yes, glycolysis, and this pyruvate can be converted to alanine, with which we are familiar here. That is transamination, which can convert pyruvate to alanine. Just to remind you, this is the reaction pyruvate plus glutamate. So that is a reversible reaction. So this is the reverse reaction of what we saw earlier, to give rise to alanine and alpha ketoglutarate. Now, alpha ketoglutarate can remain inside muscle cells, but alanine cross the bloodstream to reach liver cells. So this is alanine now in liver, and here alanine can be converted back to pyruvate. Again, the reverse reaction of the transamination reaction which we saw. So here alanine can take up alpha ketoglutarate inside liver and release pyruvate and glutamate. So glutamate reaching liver, it can give rise to, it can actually undergo oxidative deamination to release ammonia there. And what happens to the pyruvate? That is shown here. Pyruvate, it's a substrate for gluconeogenesis. Glucose can get back to muscle cells. And this cycle is actually quite similar to the Cori cycle. That also, it's an exchange between muscle and liver cells. This is pyruvate gets converted to lactate through bloodstream reaches liver, convert lactate to pyruvate. That happens only in liver, and pyruvate is obviously gives rise to glucose back to muscle cell. And there are still other exceptions. An example of serine and threonine are shown here. Now, although the nitrogen atoms of most of the amino acids are transferred to alpha ketoglutarate through transamination before the removal of the nitrogen amino group, the amino groups of serine and threonine can be directly converted to ammonia, also as shown here. Now, these direct transaminations are catalyzed by serine dehydratase and threonine dehydratase, in which pyridoxal phosphate is the same pyridoxal phosphate is the prosthetic group. The enzymes are called dehydratases because dehydration occurs before deamination. Serine actually loses a hydrogen ion from its alpha carbon atom and a hydroxide ion group from its beta carbon atom to yield an intermediate compound. This unstable compound reacts with water to give pyruvate and ammonia. Now, actually, the presence of hydroxyl groups here and here, the presence of hydroxyl group attached to the beta carbon atom in each of these amino acids. These are two hydroxyl containing amino acids, obviously. So this is the what permits the direct deamination in both these cases, in serine and threonine. Now what we have is ammonia which is released in mitochondrial matrix of liver. Now onwards we have to move to detoxify this highly toxic ammonia. And there are two methods adopted. One is the primary defense mechanism, that was formation of glutamine that we have already seen. And the secondary defense is formation of urea that take place in liver. So that we will deal in the next module.